Hello, students. Now we leave acid-base chemistry and enters the different subject, EDTA titrations. Um, uh, this is kind of a complex metry titration. Okay, and in this chapter you will learn how metal ions make complex with ligand. Okay, there are many ligands. Okay, and EDTA is one of the most famous ligand that is used for chemical analysis. Okay, yeah. First, let's consider the ion channels in the cell membrane. Okay, and in the front page of this chapter introduces very interesting thing. Okay? This ligand and metal ion complex is also found in the cell. Okay? Uh, cell membrane has a very complex structure. Okay? Usually, the cell membranes are bilayer, lipid membrane. Okay, here this lipid molecule they arranged in by layer, okay, two layers, okay, and there are many things involved in this membrane, okay, membrane proteins and a lot of things, electron transport chains, for example, and here through this membrane, ions are moving, okay, ions passing through these membranes. And the one of proteins that is responsible for potassium ion transport is chromacidin A. Okay, uh, this large, the dimer um, has this small pore, and through this pore, the potassium ions are readily pass through. Yeah, but um, the other ions like sodium ion is transport the rate is much smaller than potassium ion. Okay? People have wondered why that is. Okay? And in 2003, Dr. Roderick McKinnon, yeah, he was awarded the Nobel Prize in Chemistry for this work. Yeah. He elucidated the structure of potassium channels selectively permit potassium ion to pass through the membrane of cell-like nerve. Okay, and this is the overall figure, and this is the uh, view down ion channel. Okay, this side view, and important thing is that this part around this hole, uh, these amide groups is located. Okay, so this amide action in the channel are spaced, okay, just right to replace water of potassium hexahydrate. As you know, the most ions, both anions and cations, they have water molecules around them. Yeah? This water is called hydrate water. Okay? But in order to pass through this pore, okay, pore, this water molecules must be removed. Okay, so this pore size is just right to the this potassium ion size. So when this potassium ion is moving through this pore, these water molecules are replaced. Okay, uh, but there is little change in energy when hydrated potassium sheds uh, the water and binds inside the channel. Okay, so since this one is oxygen, it provides electron pair to potassium ion. So potassium ion in this hydrate form is usually uh, freed. Okay, so this potassium ion only can pass can be bound these amide groups here. Okay, but what about the sodium ion? Sodium ion also has six water molecules around it. Okay, for sodium ion, the spacing of this pore is too great by this much, okay, 0.04 nanometers, okay, to displace water from this sodium hexahydrate, this ion. Okay, in other words, for the sodium hexahydrate water, this pore is big, so there will be a not easy complexation of sodium ion by this amide group. Okay, so as a result, potassium ion passes at a rate of 10 to 8 ions per second. Wow, this is amazingly fast, the transport rate. Okay? This is 100 times faster than sodium ion. Okay, well, so in that sense, chromatidine 4 is selective for monovalent cations and exclude anions and highly charged cations. Okay? By the way, this chromatidine A is antibiotic, okay, made of 15 amino acids. Okay? There are 15 amino acids. Okay? Yeah, this is an example how uh, metal ion complexations are uh, occurring in nature. Okay? So in this chapter, we learn the metal and chelate complexes. Okay, the chelate, as you'll see soon, the chelate is a kind of a ligand when it forms a complex with the metal ion. The entire structure is like a ring. Okay, there are many chelating agents. Okay, but one of the most famous chelates is EDTA. So in this chapter, uh, yeah, we'll be concentrating on this EDTA, okay, its properties and how we apply the EDTA for the determination of metal ion concentrations. Okay. The EDTA is a chelating agent forming strong one-to-one complexes with most metal ions. Okay, yeah, that's the beauty of EDTA. So strong metal binding agent in industrial process and in products such as detergent, cleaning agent, food additives, and drugs. Okay, so EDTA is widely used in various areas. Okay, detergent, cleaning agent, food additives, and drugs. But in this chapter, we will consider EDTA as a very effective complexing agent, and uh, we learn how to calculate metal ion concentrations. So also EDTA has emerging player in environmental chemistry, mm, passing as unscathed EDTA complexes of toxic heavy metal ions. Okay, so since EDTA has a very high affinity toward metal ions, so EDTA can be used to remove heavy metal ions in nature, okay, in the environment. So EDTA, well, it can be right this way, but actually structure, you see, yeah, it's not exactly like this, okay. But anyway, EDTA has six, kind of arms to bind metal ions. Okay, here, blue colored the element nitrogen, two nitrogens, and one, two, three, four oxygens. Okay, they provide non-bonding electron pair two metal ions. Okay, so after forming uh, this complex with metal ions, you see this is a three-dimensional structure of manganese EDTA complex. Here the center is a manganese ion. Okay? And this manganese ion is bound by one, two, three, four, five, six arms from EDTA. So EDTA is hexavalent complexing agent, hexavalent ligand. Okay, so if the organic compounds have oxygen or nitrogen atoms, they uh, um, they have tendency to uh, form a complex with metal ions. Now, one good example is ethylene diamine. Okay, the structure is very simple. It has two nitrogen atoms in the structure, and each nitrogen atom has non-bonding electron pair. Okay, so this non-bonding electron pair now goes to the metal ion, metal cation. Okay, you know the metal ions are all the time cations. Okay, no, not anions. So after binding with this copper ion, the structure is like that. Okay, so two non-bonding electron pairs from nitrogen atoms now bind in a copper ion. Okay, this is called bidentate. Okay, bi means um, two. Dentate means this is what teeth. Okay, dent. Okay, it's like two teeth now bind the copper ion. Okay, so this EDTA is what hexadentate ligand. Okay, because there are six atoms that binds the metal ions. Okay, so metal ions are all the time, without exception, Lewis acid. Okay, metal ions are cations and they are Lewis acid. There is no exception. What is a Lewis acid? Lewis acid is electron pair acceptor. Okay. In the meantime, Lewis base is a electron pair donor. Okay. In the sense, ligand are Lewis bases. Okay. Here, look at that. This one, this ethylene diamine, you know, this molecule is a electron pair donor. Okay, electron pair, it donates electron pairs to copper ion. So this is a ligand and Lewis base, and copper ion is Lewis acid. Okay, so we say the ligand as monodentate, bidentate, tridentate, tetradentate, pentadentate, hexadentate are like that. Okay? Monodentate ligand is what? It binds to a metal ion through only one atom, like this cyanide. Okay, it provides also electron pair. Okay, but uh, it has only one, the donor. Okay, electron pair donor. So this is a monodentate. Well, naturally occurring ligands are so many. Okay? For example, ATP. You know the ATP, adenosine 3 phosphate. Okay, this ATP, this energy molecule, this ATP um, binds to divalent metal ions. Okay, very strongly, uh, such as magnesium, manganese, cobalt, and nickel ions. Okay, when magnesium ions forms a complex with ATP. Okay? This is biologically active. Here, this is structure of ATP, and ATP is what? Tetradentate ligand, because each phosphate group provides one oxygen the atom, and this one, let me see, what's the name of this part? Anyway, this one provides, this one nitrogen this atom. Yeah, so 1, 2, 3, 4. This is a tetradentate ligand. Okay, so this is three-dimensional structure. Okay? When uh, binds metal ions, so these ATP molecules now uh, what forms this three-dimensional structure, yeah, to bind the metal ions. Okay, but here also water is a good ligand. Okay, because this oxygen atom has two non-bonding electron pairs. One of them binds to metal ions. So actually here metal ions now bind are bound by four this what bond from ATP, and also there are two water molecules, although not appear here. Okay, so water, remember, water molecule is also a good ligand, but this formation constant is not good. Okay, it's very small. You will see it soon. So what is the chelate effect? Chelate is the this kind of ring shaped, uh, what when the ligand forms ring shape. Okay, that is called the chelate. Okay, for example, this cadmium. Okay, hexahydrated, cadmium ion now uh, react with ethylene diamine. Okay, the structure is like that. Okay, around the cadmium ion there are two ethylene diamine molecules are bound. Okay, here and here. Okay, so although cadmium ion has six water molecules, in this case water molecule is also a ligand. Okay, acting as a Lewis S base. Okay? But since uh, ethylene diamine has more higher tendency to bind cadmium ion when uh, binds to ethylene diamine, this water, all the water molecules are removed. Okay, here, so only naked cadmium ions are bound to what this ethylene diamine. But in this case also, there are two water molecules bound this cadmium. One is above the cadmium ion, and the other one is below. Okay, and interestingly, two ethylene diamines bind to one cadmium ion, forming. You look at that. This one, it looks like a ring. Okay, ring. Okay, you understand? So chelating effect is that the ability of a multidentate ligand to make more stable metal complexes than formed by similar monodentate ligands. Okay, I will here the equilibrium constant. We can write equilibrium constant for this reaction. Okay, that K. Yeah? This is equilibrium constant is also uh, called formation constant. Mm. This K is 8 times 10 to 9. Okay, very big. But the similar structure can be formed. Okay? That is, same cadmium hexahydrate. This ion now react with methylamine. Okay, this methylamine is simply CH3 and H2, and four methylamines bind one cadmium ion here, right? Like this. Okay, four methylamine. Okay, so around cadmium ion there are 1, 2, 3, 4 methylamine and two water molecules. But in this case, in ethylene diamine, you know, number of ligands the same 1, 2, 3, 4. Okay, but only difference between these two complexes is that this ethylene diamine here, this structure is like a ring. Okay, this is closed structure, but with the methylamine, there is no ring like a structure here. Okay, so this is called chelate. Okay, chelate. So ethylene diamine is called chelate because it forms a this one the ring like structure. Okay, and now let's compare the equilibrium constant of this one. This compound, and K is 4 times 10 to 6. Okay, wow, there is a big difference, more than 1000 times difference. Okay, so that's called chelating effect. Okay, although structure is very similar, only difference is that whether the ring like structure is formed or not. Okay, so chelating effect is that when uh, the metal ligand complex is formed, if the structure is contains this ring type structure, yeah, and then this complex is more stable. Okay, here look at that. That is called the chelating effect. Okay, at pH 12 in 2 molar ethylene diamine and 4 molar methylamine. Okay, the quotient of these two is 30. Okay, 30 at the higher pH, although this ratio becomes smaller anyway. This complex is more stable than this one. Okay. Okay, and our textbook introduces a very interesting thing, uh, which is chelation therapy and thalassemia. Okay, there is a genetic disease called thalassemia. Okay, this disease uh, is a disease on hemoglobin. Okay, as you know, hemoglobin is an ion carrier. Okay, it consists of two pairs units of alpha and beta. It has alpha and beta twins. Okay, but there are two types of thalassemia: alpha type and beta type thalassemia. Thalassemia is in Korean 지중해 빈혈증. Okay, it's called 지중해 빈혈증. thalassa here. Okay, thalassa. That is from Greek. Okay, thalassa means sea. Okay, sea actually 지중해. Okay. Okay, the beta thalassemia is a genetic disease due to deficiency of beta unit in hemoglobin. Okay, since hemoglobin is not correct. Okay, so once you have this disease, survival rate is quite low actually. Look at this figure. X-axis is age, y-axis is percentage survival. Okay, so without any treatment, okay, if you have beta thalassemia, then you know before age 10, you know almost 100%, people die. Okay, wow. So children affected with this disease can survive with frequent transfusion of a normal red blood cell. So uh, how do we overcome this disease? Well, yeah, I don't think there is a way to overcome, but frequent transfusion, 수혈, okay? If the children are frequently transfused with the normal red blood cell, survival rate increases. Okay, here look at that. This one, and transfusion only, and up to maybe age 12. Yeah, but the survival rate is good. But and then the child grows, and then survival rate drops rapidly, and maybe before 20 years, or maybe before 30 years old, right? All of them will die. Oh, that's also tragic. Okay? But there is a there is a good method to what lengthen, to increase the survival rate. That is what called chelation therapy. Okay. So problem of transfusion. Yeah? Transfusion is alright, but it's not perfect. Okay. Accumulation of 4 to 8 grams of iron per year in body. Okay. When you transfuse red blood cell, okay, the ions, excessive iron is accumulated in body. So this accumulation of iron may have effect, bad effect on the body. Okay, so the person will die eventually. Okay, because human body has no mechanism for excreting large quantities of iron. Okay, so the iron must be removed. Okay, but by what means? Okay, so most patients die by age 20. Okay, here. Okay, yeah, the iron, excess iron have toxic effects. Okay, so transfusion and uh, if you have transfusion and remove excess iron, okay, and then there will survival percentage will increase greatly. Okay, but how to remove this iron? Okay, that is that can be possible by chelation. Okay, chelating this iron, and then it can be easily excreted from the body. Okay, to enhance iron excretion, intensive chelation therapy is used. Okay? And there are many chelating agents of iron, but most successful drug is this ferrioxamine B. Okay, this one is has been known as the best, the chelating agent. Okay? So this is so powerful to chelate, trivalent iron. Okay, but this one structure is very complex, and but it can be separated from the streptomyces pilosus. Okay, this bacteria. Okay, here, yeah, this is the structure of ferrioxamine B. Okay, without this what iron, it is called this means deficient. Okay, that's ferrioxamine B. Okay, this structure is very complex, right? It is not easy to synthesize. Okay, and structure, inside structure. Here, look at that. There are 1, 2, 3, 4, 5, 6 oxygen atoms toward the inside. Okay, and ion, this one, is quite easily, and very strongly bound to these 6 oxygen atoms. Okay, now this one, this is which structure? This one, hydro hydroxamate group. Yeah, this is called hydroxamate group. Yeah, there are 3 hydroxamate groups, and each group provides 2, the oxygen ligand. Okay, yeah, this is a 3-dimensional structure of ferrioxamine E. Okay, yeah. So, but it also has a negative effect. What is the negative effect of this treatment? It's too high a dose stunts a child growth. Stunt means hinder. Okay, but and also this drug is quite expensive, and it must be taken by injection, uh, uh, not absorbed through intestine. Okay, so injection. Okay, also orally taken drug was developed. This the ferriprone. Okay, it also has a complex with ferric ion. Okay, 3 to 1.